Namaskar and welcome to CEC Gurukul Lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. In this series of lectures on analytical techniques in plant sciences, we have been discussing a lot about various microscopic techniques. We started our journey from the basic principles of microscopy, the principles of resolution and magnification. We also understood various different types of light microscope and stereo microscopes. We proceed now towards a more advanced and more efficient modification of microscope, that is fluorescence microscope. In the previous lecture, we established the basic principles of fluorescence. How any object which has the fluorescent property gets into a higher energy state and then re-emits or releases that higher state energy in the form of light of different wavelength in a fraction of seconds, less than maybe microseconds or nanoseconds, and the visible light which is emitted is of different wavelength, and this entire process is called as the fluorescence. We also established that the range of wavelength which can excite the general state of energy of a molecule is different than the range of wavelength which is emitted after or during the process of fluorescence. So there is a range of incident spectrum and there is a range of emission spectrum. And the difference between the transmission or the incidence spectrum and the emitted spectrum is called as Stokes gap, or this Stokes gap is very crucial in understanding any fluorescence microscope, because this Stokes gap is unique for any kind of fluorophore. We have also understood the basic pathway of light which takes place in any of the microscope showing fluorescence properties. So today let us continue our discussion with fluorescence microscope, and the most common fluorescence microscope happens to be epifluorescence microscope. So an epifluorescence microscope is basically a reflected light microscope in which the reflected light is with a longer wavelength than that of excitation wavelength. The illuminator is interposed between the detector and the nose piece housing the objectives. So what is an illuminator? The illuminator is a horizontal tube from where the light passes, and this illuminator is interposed between the detector. This detector could be an eyepiece or it could be an imaging camera, and this illuminator is placed between the detector and the nose piece housing the objectives. So that's the basic structure of any epifluorescence microscope. We can understand the structure of epifluorescence microscope with the help of a longitudinal anatomy. Here in this diagram you can very well see that there is a mercury arc lamp which acts as the source of light. From this source of light the light passes through the condenser aperture diaphragm, and then it passes through the field diaphragm, and then it reaches to the tube lens. So the light first initially passes through a set of diaphragms. Now in this tube lens there are certain fluorescence filter cube fitted. The fluorescence filter cube is comprised of very specific optical instruments or optical devices. This enables the filtering out of unwanted light of different wavelengths, and then this light is passed through the objective, and on the stage there is a specimen. From this specimen, which is colored with a fluorophore. Now after this fluorophore gets excited by the incident light, the radiated light belongs to a longer wavelength. It comes out, passes through a dichroic mirror, and with the help of a prism it gets reflected towards observation tube, and then here it could be observed with the help of an eye or a camera. So that's the basic structure or basic anatomical structure of any epifluorescence microscope. Rest all the structures remain common to any light field microscopy, such as the frame, such as the base, the focus knob, the stage, and other structures. In this type of microscope, the light of different wavelength, often in the ultraviolet region, is produced from an arc discharged lamp or other source positioned at one end of the vertical illuminator called as lamp house. The light from the source travels along the horizontal illuminator, and it passes through an adjustable aperture diaphragm, then through a variable centrable field diaphragm, and then impinged over the excitation filter. Now this once it passes through the excitation filter, the selected wavelengths falls on the specimen through objective lens. After getting reflected from a dichroic mirror or beam splitter positioned at 45 degree to the light path, the objective lens collects the fluorescence light emitted from the object. This then passes through the dichroic mirror and barrier filter. They allow passage of only longer wavelength light to the detector. This could be understood with the help of this ray diagram. Here we can see that there is a light source. This light source emits light and it is passed through the excitation filter. The excitation filter does not allow any other light to pass through it except the light of excitation spectrum or illumination spectrum. So the light belonging to illumination spectrum only gets passed through the excitation filter, and this gets reflected by a dichroic mirror or dichroic filter. The dichroic filter, it is adjusted at an angle of 45 degree. So the light belonging to the excitation spectrum or illumination spectrum is now directed towards the sample. Now this sample is very specifically and very meticulously stained with fluorophores. Fluorophores are those molecules which exhibit fluorescence, which show the phenomenon of fluorescence. So the sample which is stained by fluorophores, these fluorophores then re-radiate another set of wavelength of light, also called as emission spectrum, the light in the range of emission spectrum. Now this emission spectrum wavelength is always greater than the illumination spectrum, and this emission spectrum light directly passes through the dichroic filter, and it again passes through the emission filter. The function of emission filter is to stop any other wavelength light except the emission spectrum. So to an eyepiece or to an observer, only those structures will be visible which are stained with fluorophores in a background which is absolutely dark, and this creates a very sharp contrast, and thus the detailed structure of any sample is made visible. So this entire process involves three different types of filters, and all these three types of filters are referred to as special interference filters. They have an advantage of selecting lights of very specific wavelengths. In most of the fluorescence microscopes of reflected type, the excitation filter, dichroic mirror and the barrier filter are held together by means of a small block or cube-shaped filter holders. This cube-shaped filter holder is also termed as filter cube. The internal structural organization can be represented by this diagram, where the excitation light gets inside, but because of the excitation filter, only the excitation spectrum light passes through it. It goes towards the object, and from the object, after fluorescence, emission light comes. The emission light directly passes through the dichroic mirror, and this dichroic mirror then passes through the emission filter. This emission filter allows only the emission light to pass through it, and the object is seen in a dark background, and the object is seen only because of the stained fluorophore. So that's the basic pathway of light across the filter cube. Any filter cube can have these parts only. So there is a incoming light waves, and then there is a excitation filter. This has a dichromatic or dichroic mirror, and then it has a barrier filter, and this barrier filter then allows the light to move towards the fluorescence emission. So this diagram shows a typical filter cube with the excitation, dichromatic and emission filters. In fluorescence microscopy, the concentration of the light emitting fluorophores is very low. To produce a detectable level of emission, the fluorophores needs to be excited with strong intensity light sources. And the most common lamps used in fluorescence microscopes are mercury and xenon arc lamps, because they can produce a very high intensity light source which will give a higher concentration of light emitted by the fluorophores. So let us talk about fluorophores. What are fluorophores? So fluorophores are the molecules which have the ability to emit fluorescence upon excitation with light. They are also known as fluorophores or fluorochromes. Many organic molecules or compounds such as chlorophyll, vitamins, resins, butter, etc., they have high intrinsic fluorescence when they are irradiated with light. So these molecules, they naturally show the phenomenon of fluorescence. So to visualize these organic molecules such as chlorophyll, vitamins, etc., we do not have to put any fluorophore or we do not have to stain them with any biological fluorophore. So the presence of these organic molecules which show fluorescence, it is also termed as the autofluorescence. Now, of the most notable biological fluorophores which is used to stain any kind of cellular structure, green fluorescence protein or the GFP protein is the most common one, which is obtained from the jellyfish named as Echora victoria. The use of green fluorescent protein and its variants as fusion protein provides invaluable service in live cell imaging experiments. Over the past several decades, many chemically synthesized fluorophores have been used in labeling a wide range of biological systems. Such chemical compounds often have ring structures with conjugated double bonds. Similarly, in case of GFP, if we look at the architecture of GFP, you will find that it's a conjugated protein, beta barrel and beta sheets, and in the center of these structure, the chromophore molecules, which are generally ring structures or cyclic organic structures, they are found, and because of the presence of these cyclic structure, the GFP shows fluorescence phenomena. So that was about the fluorescence microscopy. Now we shall move from fluorescence microscopy towards another very important variation of microscopy, that is called as confocal microscopy. So confocal microscopy, or the modern day terminology used for confocal microscopy, is confocal laser scanning microscope, CLSM. So this has helped in creating sharp images of a specimen that would otherwise appear blurred when viewed with a conventional microscope, and thus has amounted to widespread applications in recent years. So what is a confocal microscopy? What is a confocal microscope, and what are the basic principles which are applied in confocal microscopy? Let us try and explore this aspect. So as far as the confocal microscopy is concerned, the structures which we use is nothing but a modification of fluorescent microscopy, where, so you can understand this with the help of a diagram where a pinhole is used to focus the light, and by using a pinhole, the focused light is incident on a specific plane. So the diagram shows a basic light pathway of the confocal microscope. Here, a light source, usually a laser beam, is passed through a pinhole. This pinhole allows the light to be focused at a very specific location, at a very specific point. As we move this structure, the image is captured with the help of a detector, and as we move our object or our specimen, the image is collected again and again, and all these collected images, they are superimposed to create a sharp and almost a three-dimensional structure of any object. So with the help of just by adding a pinhole, we can look into any plane of the object. So the principle of confocal microscopy employed in all modern day confocal microscope was discovered in 1955 by Marvin Minsky. The desire towards the development of confocal approach was to image biological events as they occur in living tissues. The earlier versions of confocal microscope was discovered by Minsky, which used zirconium arc as the point of source rather than laser beams. A point of light is focused across a specimen sequentially and then through collection of some of the returning rays. A point-by-point image is constructed. To maintain sensitive alignment of moving optics, the specimen is scanned by moving the stage rather than the light rays, and the image is gradually reconstructed through collection of the remaining desirable light rays by a photo multiplier. Here in this diagram you can see that the image is formed at a particular axis, and these images at different planes are created, and we move the stage, and all of these planes, they create different images, and these images are then superimposed frame by frame to create a three-dimensional, much contrasting and sharper image. The key principles of Minsky's conventional design of confocal microscope have been maintained in the modern confocal microscopes also. A laser is used as a light source instead of a tungsten or mercury lamp, and additionally a sensitive photomultiplier detector and computer controlled movement of scanning mirrors and other scanning devices facilitates the collection and display of images. So the basic outline diagram of a confocal microscope can be understood with the help of this diagram, where from a light source the laser light is being focused at a particular point because of the presence of an illumination pinhole, and they are reflected back, and there is a confocal pinhole, so it blocks any extra or the noise in the image produced, and the detector captures the sharp pinpoint image. These pinpoint images are created in multiple numbers with the movement of stage, and each movement or each point which is being moved by the stage, there is a respective image which is created, and these images are superimposed frame by frame to produce a three-dimensional structure. So the idea of point-to-point illumination of the specimen and rejection of out-of-focus light have been incorporated into focal microscope when we compare it with any other light microscope or light field microscope. In contrast to the conventional microscope where object-to-object transformation takes place simultaneously and parallel to all objects. In case of confocal microscopy, the specimen is irradiated in a point-wise fashion in a serial manner, and the specimen detail irradiated is measured point by point as well. So this the comparison between a normal microscope and a confocal microscope. In a normal microscope, the image is created and the object is illuminated and image is created simultaneously, whereas in case of a confocal microscope, the light source is passing through a pinhole and it creates a sharp image and it is being received by the detector, and the other pinhole blocks any out of focus light. So whatever image is being captured by the detector happens to be very sharp image, and multiple such sharp image at different planes, they are superimposed to create a very contrasting and a very clear picture. So the distinguishing features of any confocal laser scanning microscope is the confocal aperture, also called as pinhole. This pinhole is arranged in a plane conjugate to the intermediate image plane and hence to the object plane of microscope. And the PMT or the photomultiplier thus detects the light that has passed through the pinhole. The pinhole also helps in rejecting out of focus light rays, that is excluding the light coming from the object points outside the focal plane and eventually from detection. However, the optical sectioning potential depends strongly on the size of the pinhole. So this can be understood with the help of the diagram, where the size of the pinhole the optimal size of the pinhole will determine the precise image of the object. So if the pinhole is very large, then also the sample will be out of focus. Another important component of confocal microscope is the photodetector that helps in capturing the light from the specimen. The measurement accuracy is enhanced by increasing the number of photons arriving at the detector, which is achieved either by increasing the intensity of the fluorescence signal or averaging data from many frames. So pinhole reduces the intensity of emission reaching the detectors. The microscope illuminates specimens using a laser beam focused by an objective lens down to a diffraction limited spot, and the position of the spot is controlled by scanning mirrors which allow the beam to be swept over the specimen in a precise pattern. So this is how the entire process in a confocal microscope takes place. I hope I have been able to give you a broad understanding of the basic principles and functioning of fluorescence microscope as well as confocal microscopes. Thank you so much for your patient listening.